So in the last class, in continuation of our discussion on Introduction to Bioinformatics, we discussed about the different important areas under bioinformatics. And also we have discussed about how bioinformatics can play a very important role in solving some of the problems related to our understanding of biological complex phenomena. So we took the example of finding genes in a genome sequence and how different bioinformatics tools were developed for identification of genes in a long sequence of a genome, because most of the part of the genome in any organism is occupied by non-coding sequences that do not have any relevance to the genes or the functional characteristics. It is very difficult to find out the coding sequences or the gene sequences within a genome sequence by manual reading. It is not possible. So bioinformatics tools have played a very important role in identification of the genes and the corresponding regulatory sequences in a genome, and thereby made it possible to annotate the entire genome sequence to find the role of different segments of the sequence in the functional characteristics of the particular organism. So today we shall continue in that process, and we shall discuss about some of the other issues in which bioinformatics has played a role in understanding the complex biological phenomena. So this is one area where the question is how to compare two sequences of either two organisms or different strains of the same organism, how we can compare them and find out what are the variations at the genomic level or even at the proteomic level. That means how can you best compare the sequences of genes and genomes, or for that matter, the amino acid sequences of the proteins? What are the basis or the criteria based on which they can be compared? And we can say that to one particular sequence is more alike or identical to another sequence compared to a third sequence. So some other people are trying to enter. Let me admit them. So in this process also, the bioinformatics tools can help us in finding the comparable sequences among sequences, and by that process to establish relationship among sequences and for that matter among organisms based on their sequence similarity, and for that matter based on the similarities in the evolutionary process. So in the process of phylogenetic analysis, this plays a very, very important role. So in order to do so, we have to first define a set of possible criteria based on which we can compare the sequences. For example, in the case of genetic variation, as we know, there are two major causes of variation. One is the mutation, and the other is the genetic recombination. Now, taking mutation as a method of variation in gene or genome, I think there are two types of mutations. That is one is the point mutation, which occurs due to replacement of one nucleotide by another. And the other is called frame-shift mutation, where there is an insertion or deletion of one or more nucleotides from a gene or genome sequence. Now, this can be probably happening at any point of time in the evolutionary process. Of course, point mutations normally occurring in a more frequency compared to the frame-shift mutation. Now, these phenomena of either point mutation or frame-shift mutation, replacement or deletion or insertion, can be translated into certain mathematical models, because these nucleotides represented by ATGC, these 4 letters, can be considered as discrete characters in the computational method, and a change in character or replacement that will represent the point mutation or insertion or deletion of characters within a sequence can be converted into numerical or mathematical expressions by use of the bioinformatics tools. And thereby, we can also calculate by using the arithmetic or mathematical process the difference or the distance between 2 sequences or the similarity between sequences in terms of numerical values. And that is the usual process of comparing sequences, what we refer to as sequence alignment. We will go into detail about sequence alignment in some later class, but today I shall only give you a preliminary idea how sequence alignment can be done and how we can mathematically define the distance or similarity between sequences. Take the example of these 2 sequences, say sequence 1 and sequence 2. So say these 2 sequences are from 2 strains of the same bacteria. Let us take it like that. Now, they represent the same gene, but if you compare them one with the other by comparing each of the nucleotides separately, starting from the nucleotide 1 in both sequence 1 and sequence 2, if you arrange them like this, then if you compare nucleotide to nucleotide, we find that there are some positions in which the nucleotides match, and in other positions the nucleotides do not match. So let us represent the matching nucleotides with the help of these perpendicular lines. So suppose there are around 30 nucleotides here. Out of 30 nucleotides, how many nucleotides are matching? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 14. So out of 30, only 14 nucleotides are matching to each other. So in terms of mathematical similarity between these sequences, we can simply say that there is a similarity level of 14 by 30 multiplied by 100, means less than 50% similarity, almost around, say, 48% similarity between the 2 sequences. So if we compare this by this way, this is a comparison between sequences based on similarity or matches in the nucleotides, but this does not represent the real biological situation in which one sequence deviates from the other in the evolutionary process. So we shall have to keep in mind that these deviations, as we have discussed earlier, basically are due to 2 types of mutations. One is the replacement mutation or point mutation, and the other is frameshift mutation or insertion and deletion. If we take into account those biological events or evolutionary events into consideration, then each of the mismatches will represent 1 point mutation. But of course, here it does not, in this type of comparison, it does not represent the other type of mutation, that is your insertion or deletion. So what we do by taking into consideration these evolutionary events, we can arrange and rearrange the sequences by comparing to each other in different ways by putting certain gaps in between so as to get more matches or similarities following putting the gap. Say, for example, here up to the nucleotide T, that means 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Up to the 11th nucleotide, we have kept it as it is because these 2 differences in nucleotides T to A and A to Z prior to this 11th nucleotide, one is at the 5th position and the other is at the 8th position, can be explained by the evolutionary phenomenon of point mutation. T is replaced by A, this is a point mutation. Z is replaced by Z, A is replaced by Z, this is another point mutation. We can explain it like that. But following this 11th nucleotide, T, here you see 4 nucleotides are different from each other. 4 nucleotides are different from each other. But if you see here, C to T, there is a variation. But after that, CAZ, if you consider these triplet, here CAZ is there, even it is followed by T, T, the rest, many of the nucleotides are same. So if we consider that, instead of a point mutation occurring here, if we consider that there is an insertion mutation occurring here or a deletion, then we can represent it by replacing with a gap. So this gap means a deletion. So when we introduce a gap here, it will represent that in this particular sequence 1, there is a deletion of 1 nucleotide, or otherwise the same thing can be expressed as an insertion in the sequence 2 by T. So if we consider that it is the real event occurring in the evolution by which these two sequences have differed from each other from this point onwards, then you see matches are increasing. Now here in the previous position, matches were very few thereafter. But after adding this insertion here, a gap here, you see there are 6 nucleotides which are matching. Then by this way, if we put one or more gaps thereafter to increase the number of matches in nucleotides following that particular insertion or deletion, then we find different kinds of arrangements of the two nucleotide sequences. So this kind of arrangement or rearrangement can be done in many different ways, but our idea is to find out the best matches. That means after arranging the sequences one upon the other by putting gaps here and there, one or more, we shall have to find out of hundreds of such arrangements, in which arrangement we get the maximum matches. So that is the original situation in the two nucleotides before evolutionary changes could have occurred. So now, in these cases, because we took only 30 nucleotides, this arrangement could be made manually. I could make it manually by reading. But this is not possible to make if thousands of nucleotides are concerned. If in a gene sequence or genome sequence, many millions of nucleotides are involved, this kind of arrangement done in hundreds of times, finding out the best matches, cannot be done by manual process. There we take the help of the computer. So what the computer will do, computer will take each nucleotide as one character, and it will try to put gaps randomly here and there, one after the other, more than one gap also sometimes. And every time it puts gap to rearrange the two nucleotide sequences, it will calculate the number of matches thereafter. So then in the particular arrangement in which maximum matches are found, maximum identity is found, that is usually taken as the alignment of the two sequences. So that is not a simple comparison that has taken into account the evolutionary events by which the two sequences may differ from each other. So by this way, a particular arrangement is reached, and that will be called as an alignment. One sequence is aligned with the other, and the criteria of variation is based on the evolutionary variation between the two sequences occurring by point mutation or by frame shift mutation. Now, after we do this kind of alignment or rearrangement of the sequences, wherein we find the best matches between the two, now we can go for calculating the variation between the two sequences, either in terms of distance, how one is distant from the other, or in terms of similarities. We can express in terms of distance or in terms of similarity. Now, why this is so? Because you see, these variations could have occurred in the process of evolution at some time in evolutionary history between these two sequences, although they were supposedly the same identical sequences at one point of time in the evolutionary history. So suppose these two sequences were exactly alike sequences, similar sequences in the evolutionary history at some point of time. Now they differed subsequently in subsequent generation because of insertion, deletion, or by point mutation occurring in the sequences. So that can be explained after the alignment of the sequences. Here there is a T2A replacement by point mutation. Here there is an insertion or deletion in one sequence, or maybe insertion in the other. Here again, 3 insertions at the same position or 3 deletions at the same position, a frame shift mutation. And here again, at 2 positions consecutively, there are replacements or point mutation. Again, in the last nucleotide, there is a insertion in one or deletion in the other. So by this way, you can explain in terms of the evolutionary phenomenon. So this is the final sequence alignment. Now we can go for calculating the variation between the 2 sequences, either in terms of distance or in terms of similarity, by using certain mathematical scale of measurement or a model. A model scale of measurement has to be first decided. Say, for example, while finding out distance between the 2 sequences, let us, for simplicity, take that when there is a match, as there is no distance or there is no difference between the 2 sequences, when there is complete identity in terms of one position, suppose in first position Z to Z, both the nucleotides are same, there is no mismatch or there is no variation. So we can say that distance is 0. So while counting distance, a match will score as 0. Then when there is a substitution or replacement of one nucleotide by another, then there is a distance. One difference has taken place by way of point mutation. So let us give it a value of 1, a distance value of 1. Then while there is a insertion or deletion, as insertion deletion is an evolutionary phenomenon which occurs less frequently than that of point mutation, therefore this kind of distance will happen only in longer duration of evolutionary time. And therefore, if we find this distance, the weightage of distance will be more. So let us take the score of 2 for every insertion deletion. This is a simple scale that we have decided to differentiate between the 2 sequences. So now for every match, we will put a score of 0. Sorry. For every substitution or replacement, we will put a score of 1. And for every insertion or deletion, we will put a score of 2. So if these are the criteria of measuring distance between the 2 sequences, then we can score the distance score. We can find out the distance score of the 2 sequences. How? Now, the matches have got no score. Therefore, we will not count how many matches are there. Every substitution has 1 score. How many substitutions are there? Here, there is 1. This is another 2. Then 3, 4. So 4 matches. So 4 replacements or substitutions are there. 1, 2, then 3, 4. 4 distances are due to substitution. So total distance score will be 4 into 1 is equal to 4. Then how many insertion deletions are there? 1, 2, 3, 4, 5. So for each insertion deletion, our scale is 2, score is 2. So for 5 insertion deletion, total score will be 2 into 5 is equal to 10. So therefore, the total distance score will be 14. Okay. So we can give a numerical value to the distance. We can say that distance score between these 2 sequences is 14. So if you consider it as reverse of similarity, so that means out of 30, if everything is same, identical, all nucleotides are identical, then I think there will be no distance between the 2. So here the distance score is only 14. Now, if you consider the similarity between the 2 sequences, on the other hand, the scoring matrix or the criteria for scoring will be different. But if we calculate similarity, then every match is important because a match indicates a similarity. So let us take 1 as a score for every match. Then for similarity, substitution has got no score because when it is substituted or replaced by another nucleotide, that means in that position, there is no similarity. So similarity score will be 0. Then insertion deletion indicates more dissimilarity between the sequences, and therefore similarity score will be negative. So let us take it as minus 1.5. If you take this particular scoring matrix for measurement of similarity, the same two sequences can be also aligned, and the similarity score can be measured by using this particular scale. Now, how many similarities are there? 4, 5, 6, 7, 8, 9, 15, then 21, 24. Total 24 perpendicular lines are there, means 24 positions. There is complete match. Therefore, matching score will be 24 into 1, 24. Then here now replacement will not be counted. Only insertion deletion will count. 5 insertion deletions are there. Each insertion deletion score is minus 1.5. Therefore, total insertion deletion score will be minus 1.5 into 5 is equal to minus 7.5. So total similarity score will be therefore 24 minus 7.5 is equal to 16.5. So similarity score is higher than that of distance score. Means these sequences are more similar to each other compared to distance. Means their similarity score is higher than that of distance score. So this is 16.5 similarity score, but the distance score was 14. So this is the kind of measurement that we can do by applying a particular scoring matrix. But the scoring matrix should take into account all the evolutionary events that can happen, which can deviate one sequence from the other, how variations occur between sequences, and therefore this cannot be so simple as we have taken here for an example. We shall discuss about the scoring matrices, how they are designed, and what are the standard scoring matrices for nucleotide sequence alignment, or for that matter, amino acid sequence alignment. There are different scoring matrices already standardized, and they are being used, and how they are made, what are the criteria or the logic behind development of such scoring matrices. We shall discuss in detail in later classes while we're discussing about sequence alignment. I think at this point of time, at least you could understand how bioinformatics tools can help in measuring distance or similarity between sequences, so that we can find out the relationship between sequences, and for that matter, relationship between species of organisms. Is it clear to you?
Yes, sir.
Yes, sir.
Okay. Now, another area of complexity for biologists was the protein structure prediction. As you know, protein is the structural and functional unit of life. The entire bodily structure is composed of proteins, and also all the functions of a living system is dependent upon protein. And the functionality of proteins are dependent upon the overall three-dimensional structure of the protein. The protein structure is a complex structure. As we know that there are 4 hierarchical levels of protein structure. I think there should be a slide here to explain it. Let me find it out. Okay. You know that there are 4 hierarchical levels of protein structure. First one is the primary structure, which is composed of amino acid chain, a sequential arrangement of amino acids. Then secondary structure, when the amino acid chain undergoes some kind of folding, it starts developing structures of the secondary structure, beta sheets, alpha helices, coils and loops, and that constitutes the secondary structure. So after the secondary structure is built, there will be further folding and joining between different folds because of different kind of intermolecular interactions and bone formation, and therefore it will take the tertiary structure, which is a 3-dimensional structure. And in a particular protein, there can be more than one tertiary structure or more than one domain. And finally, all the domains together in 3D model will develop the final quaternary structure. So these are the different levels of protein structure. Now, the quaternary structure is built based on the variation in the tertiary structure. If the tertiary structures are different, then the quaternary structures will also be different. Then tertiary structure is built on the secondary structure, because the way the secondary structures are built, after getting it folded more and by bone formation between the different components and the folds, finally the tertiary structure is built. Similarly, the secondary structure is built based on the primary structure or amino acid sequence. So therefore, if we correctly know the association of the primary structure or the amino acid sequence with different components of the secondary structure functionally, then we can be able to predict the secondary structure based on the primary structure. Similarly, based on the secondary structure, it may be possible to predict the tertiary structure and so on. So this is the basis of prediction of protein structure by bioinformatics tools. That means if you have got with you the basic minimum amino acid sequence of a protein, if you do not know what is the functional quaternary structure or the three-dimensional structure in the native model in the particular organism which is working, if it is not known properly, even then the bioinformatics tools can predict the three-dimensional structure based on the information that we have on the amino acid sequence alone. Now the question is, why this is essential for us? Why the protein structure prediction is necessary? You see that a particular organism has got thousands of proteins within its body. Say, for example, although human beings have got around 22,000 functional proteins, as functional genes, but there are more than 1 lakh different proteins that we discussed earlier. So all these 1 lakh different types of proteins are having different three-dimensional structures. Of course, proteins are also divided into protein families. Means based on the structural similarity, they can also be similar in terms of function. So functional similarity among proteins may be due to their three-dimensional structural similarity. So configuration of the protein is related to its relevant function. So they can be divided into different families based on their structure. As we know that we can classify proteins based on their physicochemical properties, say negatively charged, positively charged, or say even the amino acids for that matter can be divided into different classes or different groups based on their properties. Say for amino acids can be negatively charged or positively charged. It can be aromatic or aliphatic. It can be acidic or basic, or it can be sulfur-containing amino acid. It can be variable in terms of their physical and chemical structure. Sorry, physical and chemical property. So this property is also reflected on the final protein structure, and the final protein physicochemical property is based on the types and composition of the amino acid which constitute a particular protein. We shall discuss about all these things in greater detail, how protein structure prediction is achieved in later classes by using the different 3 methods available for protein structure prediction. But what I was discussing about why this protein structure prediction is necessary. You see, the structure of protein is related to the function. Therefore, to elucidate the specific function or to find out similarity in function between proteins, we must have to have the three-dimensional structure known. To determine the three-dimensional structure of proteins, we have got basically 2 different processes. One is, or methods available. One is your NMR spectroscopy, and the other is X-ray crystallography. So both these physical experimental techniques can give rise to the protein structure, experimental models of protein structure, which are more accurate than prediction of structure by bioinformatics tools. But the fact is that these two methods of protein structure determination by experimental method are having certain disadvantages. Means they cannot be done in all circumstances. Say, for example, for X-ray crystallography, the proteins are to be first crystallized. Unless they are crystallized, X-ray diffraction method cannot be used for determining the protein structure. Similarly, for using the NMR spectroscopic method, the proteins are to be in soluble form. So all proteins are not soluble, and all proteins are not capable to be transferred to or transformed to the crystal form. And therefore, the determination of protein structure by these two methods are very, very complicated and limited. And that is why so far only few thousand protein structures have been made available by X-ray crystallographic or NMR spectroscopic methods. So as a result, the structure is not available for study, and therefore we cannot use those available structure for determining the protein function if they are not available in experimental structures given by X-ray crystallography or NMR spectroscopy. So as only few thousand such structures are available now, so our understanding of protein structure, and for that matter protein function, is limited. So to enhance our understanding of protein function, we must have to have more numbers of proteins in terms of their three-dimensional structure. So that is possible now by use of computational method, by use of in silico bioinformatics tools, by which, if not structurally determined experimentally, a protein can be predicted for its three-dimensional structure if at least the primary structure of amino acid sequence is available. So I'll not explain how this protein prediction is possible by use of the variation in the primary structure. We will discuss it later on while taking on this particular chapter. So I'm just skipping some of the slides. Okay, we'll discuss it later on. Now, there are three basic methods of protein structure prediction using bioinformatics tools. One is called homology modeling. Second one is threading. And the third one is de novo protein structure prediction, or ab initio method. Ab initio method. So these three methods are available. So in homology modeling, we first go for determining the similarity between sequences by doing sequence alignment between the two amino acid sequences of the proteins. Suppose we are to do homology modeling between two proteins based on their primary sequences or protein sequences, then first of all we will go for finding out the sequence similarity. Okay, so now if two different proteins are having very high level of sequence similarity, they are homologous for most of the length, then we can assume that they are also functionally similar. If one is suppose amylase, a carbohydrate degrading enzyme, then the other one will also be an amylase because they do have high level of structural similarity. So that means based on the homology between sequences, if one is known, then the function of the other can be predicted. So in a nutshell, homology modeling represents the determination of the protein function or prediction of the protein function based on a prediction of structure of a protein by finding out sequence homology. So if two sequences are similar in terms of amino acid sequence, we can predict that their three-dimensional structure will also be similar. So based on the known three-dimensional structure of one protein, the other homologous proteins can be predicted for their three-dimensional structure. So this process is called homology modeling. Then second one is threading. So in this case, we go for the protein classification into families. Proteins which are similar in their physicochemical characteristics can be classed into or grouped into specific families. So if a particular unknown protein belongs to a particular family in terms of their physicochemical properties, then we can predict that their three-dimensional structure will also be similar. So if it belongs to a particular family of protein, if the three-dimensional structure is not structurally determined by X-ray crystallography or the NMR spectroscopic method, we can predict the structure of this unknown protein if the structure of some other related proteins in the same family are known. So given a sequence of amino acid, we can select the family to which the given sequence may belong to. And when we know that a particular unknown protein belongs to a particular family, its structure can be predicted based on the known structure of other proteins in the same family. So this is called the process of protein structure prediction by way of threading. Then if there is no sequence similarity and also we do not have sufficient information to classify unknown protein into a specific family, these two processes of protein structure determination cannot be used. In that case, we shall have to start from scratch. So that process of protein structure detection or prediction based on the physicochemical properties of proteins based on physical principles rather than based on previously known structures is called as de novo protein structure prediction or ab initio method. This is much more complex and it depends upon certain physical phenomena. So principles of physics and chemistry are used here to develop certain algorithms based on which the protein tertiary structure is determined by using certain mathematical models. So this particular process of protein structure prediction is called as de novo protein structure prediction or ab initio method. So we shall discuss about all these methods in little detail in the later class. So in 1965, a Nobel Prize in Physics was won by a scientist known as Richard Feynman, who said, if we were to name the most powerful assumption of all, which leads one on and on in an attempt to understand life, it is that all things are made of atoms and that everything that living things do can be understood in terms of the jigglings and winglings of atoms. So he predicted that like the material world, probably all biological complex phenomena can also be understood if we can understand the intermolecular or interatomic interactions occurring within the biological system. So now this particular statement of the Nobel laureate has come true because today we are using more and more principles of material sciences like physics, chemistry, mathematics, statistics, engineering, etc. to understand the interactions occurring at the molecular level within biological system so that we can understand the biological functions. So we shall discuss in due course the various sort of steps or methods in which the physical and chemical principles are being used to study the intermolecular interactions, to correlate these interactions with biological function. So there are many different in silico methods available to predict the intermolecular interactions, and therefore these interaction studies may lead to understanding of the functions of different biological molecules, and also this knowledge of intermolecular interactions can be utilized for understanding between a drug, understanding the interaction between a drug and its target, to determine and identify the drug molecules or the target molecules in a particular disease, and also for many other biological activities. So we shall discuss them in detail in future. So thank you very much. So I conclude for today. So this is the introductory class on bioinformatics that we ended today. In the next class, we shall discuss about the biological databases. So thank you very much for joining me in this class.